All right, so moving on to our third lecture in fat synthesis or fat metabolism. Our first lecture kind of dealt with dietary fats and thinking about how we can even get fatty acids into our cells, whether they were from an endogenous source like adipose tissue or an exogenous source like dietary fats. Our second lecture dealt with the metabolism of those fatty acids. So once you get those into cells, beta oxidation is the process that kind of breaks those down. And then also dealing with ketone body synthesis. So the liver is able to make acetyl-CoA from the breakdown of fatty acids, but it's not able to deliver that to peripheral tissues as acetyl-CoA because there's no export of acetyl-CoA. However, you can convert acetyl-CoA into ketone bodies, which can be exported and are a water-soluble source of fuel that can be used by many peripheral tissues. So in this chapter, we're going to move on, or in this section of this chapter, we're going to move on to fatty acid biosynthesis. So we'll do a lot to contrast the biosynthesis route with the beta oxidation route. And again, we'll kind of set up a couple of things to think about regulation of this process, but we won't deal with that until the very end of this chapter. So remembering that nature's most efficient form of stored energy is fatty acids, when we have excess fuel that's metabolized to acetyl-CoA, it doesn't matter what the source was. So carbohydrates, proteins, or lipids, everything's funneled to acetyl-CoA, and if we're not in a need state for energy, we're going to take that acetyl-CoA and assimilate it into fatty acids. This happens to be the C16 fatty acid palmitate, which is a saturated fatty acid. Now we can see why we needed the pentose phosphate pathway to do its job to generate the NADPH that we need as reductive biocurrency for this process. And again, once we make these fatty acids, we'll make them into triglycerols, and then we'll be able to store them in our fat cells. So part of our activity, again, is going to be really contrasting these degradative versus synthetic pathways. And a couple of highlights to point out is, first and foremost, we have a difference in location. So beta oxidation occurs in the mitochondrion, biosynthesis of fatty acid occurs in the cytoplasm. So we have to think about transport, and we'll deal with that in just a moment here. But another thing that differentiates fatty acids that are tagged for beta oxidation versus fatty acids that are tagged for synthesis in being made into triglycerols is the presence either of an acyl carrier protein, which we haven't yet talked about, that's in this chapter, or again, fatty acids are tagged with CoA to make them into that high energy fatty acyl-CoA. That's a substrate then for beta oxidation. So fatty acids that are going to be synthesized into triglycerides are tagged with this ACP or acyl carrier protein. And as we'll see, that acyl carrier protein is actually part of the fatty acid synthesis complex, so fatty acid synthase. So there's many reactions that we'll talk about that actually will synthesize these fatty acids, but they're all part of a multi-enzyme complex. So we'll see that in just a few slides here. So again, part of what we'll do in our activity is kind of contrast oxidation in biosynthetic processes, contrasting them by their cellular location, what's tagged, what the fatty acid is tagged with, who kind of the redox players are. Stereochemistry, we're not going to talk too much about. And then again, we're going to see an interesting new molecule here as a molecule called malonyl CoA. So we'll look at the structure of that and understand how that's an important player in this biosynthetic route. So again, first and foremost, we need to think about the fact that acetyl-CoA, being a really, really important branch point metabolite, really does not have transporters in any cellular membranes to any appreciable extent. So not only can acetyl-CoA not be exported out of the cell, acetyl-CoA can't even be exported between parts of the cell. So acetyl-CoA is generated in the mitochondrion by lots of oxidative metabolic processes. But again, fatty acid synthesis is going to occur in the cytosol. So how we think about doing that is we're going to see transport systems like we've seen before. We have to hide this molecule as something else. So we kind of begin the first step of the citric acid cycle using citrate synthase to condense acetyl-CoA into citrate. That's going to be exported out into the cytosol through this tricarboxylate transport system. We've actually talked about that before, and we'll review in our activity kind of the effects that citrate has in regulating the activity of other pathways. Once we're out into the mitochondrion, we can have this ATP citrate lyase that's going to regenerate this fatty acid that can be used as a substrate for fatty acid synthesis, and then regenerate oxaloacetate on the cytosolic side. So again, the tricarboxylate transport system is very useful for that. And then again, that oxaloacetate needs to be returned to the mitochondria. Remembering that there's not any transport systems for oxaloacetate, we see the introduction of another transporter that we talked about in our gluconeogenesis chapter, that there are transporters for pyruvate. Okay, not going to go through a lot of details here because this will be part of our in-class activity, but we're going to review again carbon-carbon bond formation and then carbon-carbon bond cleavage. And in this case, carbon-carbon bond formation is important because when we're making these fatty acids, we have to make carbon-carbon bonds between these acetyl-CoA molecules. So we'll review kind of the Claisen condensation chemistry, which was actually part of our activity from last class. Deprotonation of the alpha carbon on an ester generates a nucleophile, and we'll have another ester that's going to be an electrophile, generating a tetrahedral intermediate that now has a good group, and having an ester here will generate a beta-keto ester. And so again, this beta-keto ester will be then further modified to generate our fatty acids. So one of the things that's important to think about is, flipping back to this, we really don't tend to see this chemistry happening very easily in biological systems because of the fairly neutral pH. So when we think about making carbon enolates and making biological enolates, decarboxylation is a very common way of doing that. And so as part of our activity, we'll kind of review chemistry from our gluconeogenesis chapter where we converted pyruvate to oxaloacetate. And that pyruvate carboxylase chemistry, remember, is what's going to add that CO2 to pyruvate to make oxaloacetate, which is really kind of a hidden pyruvate enolate, if that makes sense. Again, we'll go through that in our activity. But we're going to add CO2 to acetyl-CoA in much the same way that we add CO2 to pyruvate, and generating a beta-keto ester, or in the case of malonyl CoA, a beta-keto thioester. And that's going to allow us to be able to generate our nucleophile and do our chemistry. So again, malonyl CoA is really kind of a hidden acetyl-CoA thioester in disguise. All right, so first thing we need to think about, just like when we talked about activation of fatty acids for beta oxidation, we have to think about activation of fatty acids to do some condensation chemistry, and that involves converting them to malonyl CoA. So we're going to see chemistry very similar to what we saw with pyruvate carboxylase. So a vitamin B7 necessity, so we're going to have biotin that's going to allow us to have that transferable carbon dioxide. And need to do some activation here, so it's going to need to use a little bit of ATP. And we're going to add CO2 to acetyl-CoA, generating what we call malonyl CoA. So it's a carboxylated version of acetyl-CoA that can be easily decarboxylated to allow us to generate the nucleophilic species that we need to do condensation chemistry. So this acetyl-CoA carboxylase is the enzyme that's responsible to that. We'll refer to it as ACC. ACC is a really, really important enzyme because this is the rate-determining enzyme for fatty acid synthesis. So we'll review some chemistry we've seen already, where we have vitamin B7, for example, required for pyruvate carboxylase. So a theme, again, that we're going to see recurring here, we want to couple things together and we want to make carbon-carbon bonds. It's not uncommon that we carboxylate something so that we can decarboxylate, generate our carbon nucleophile that we can then do chemistry with. So not too much will be spent in this section, in this lecture, talking about the regulation of ACC, but it is subject to both allosteric and covalent modification. Not surprisingly, phosphorylation of this will inactivate this. When we're synthesizing fats, that's because we're in a fed state, so we would expect phosphorylation to when we're phosphorylating things, we're in a need state, so we would not expect phosphorylation to activate this enzyme. We'll talk about a lot of hormones that we've already spent some time talking about and thinking about how they regulate this process. But again, that will be our next lecture. All right, so I'm going to introduce this here, and then we'll talk about the details of the steps in our activity. I'll just highlight them in the next few slides. So fatty acid synthase is a multi-subunit enzyme that has all of these different domains here that allow us to do the chemistry, these multi-step chemistry that we see to synthesize fatty acids. So when we talked about using this acyl carrier protein to tag fatty acids, that's actually part of this fatty acid synthase. So we're going to have this thiol group that we're going to do a lot of these trans thioesterification reactions to take the acetyl-CoA group and kind of put it onto this thiol. This is going to be another important thiol that we'll see, not again in a pantethene group here, but as a cysteine that's going to be able to generate that thioester. Fatty acid synthase exists as a dimeric species, so we're going to have two of these kind of come together to facilitate chemistry. We're not going to talk about the details of any of these specific enzymes. They're kind of referred to on the slides, but it's not anything that you need to know. I want you to know that fatty acid synthase is a multi-subunit enzyme. Acyl carrier protein containing this thiol group kind of in this long pantethene arm is one part of it, and then we've got an important cysteine residue. Okay, so we're going to walk through this fairly quickly because I want to leave the details for our activity. The first step that we need to do is we need to do what's called priming, which is putting these CoA flavors onto the acyl carrier portions. And so again, we've got malonyl CoA and acetyl-CoA, and then we're just going to do a trans thioesterification reaction to get them onto their respective acyl carrier proteins. And again, highlighting the fact that we're doing this thioester swap because if we've got CoA on groups, that means we're going to have these being tagged for beta oxidation, and they should be in the mitochondria. If they are tagged or part of the acyl carrier protein, that means we're doing fatty acid synthesis. But you can see the ends of these molecules are very similar, having this cysteamine group. Okay. Condensation. So really what happens here is we've got our acetyl-CoA that's now acetyl ACP. We're going to take that acetyl group and we're going to put it onto that thiol in that one enzyme that was called Ks. Don't worry about that. But now we've got this acetyl group as a thioester actually within our fatty acid synthase enzyme. We're going to have malonyl CoA that's still attached to its acyl carrier protein. In situ, it's going to decarboxylate, generate that nucleophile, react with this carbonyl here, liberating this enzyme in its free state, and then attaching it there. So let's take a close-up here, again, at the details of this. Decarboxylation of malonyl attached to the acyl carrier protein. Liberates CO2, generates our reactive nucleophile. It's going to attack this acyl group, this acetyl group that's part of this Ks enzyme. It's going to liberate the free cysteine, and it's going to attach those two carbon pieces together. So we're going to end up with essentially the net of two acetyl-CoA groups coming together. We'll go through the details of that in our activity if that was kind of quick there. These next three steps should look familiar in terms of they are the reversals of what we saw in beta oxidation. So again, if we start down here, beta oxidation saw an oxidation to an alkene, hydration to an alcohol, and then oxidation to a carbonyl. We're doing the reverse of this. We're going to do a reduction to an alcohol, a dehydration to an alkene, and then another reduction from an alkene to an alkane. And importantly, the reductive species that we see here are exclusively NADPH. Okay, so again, that should be chemistry that's fairly straightforward. And the idea is we're just going to kind of repeat this process until we have a fatty acid that's the length that we want. And it's typically somewhere between 16 and 18 carbon atoms. So again, keep repeating this process, or in case repeating 6 rounds of that will allow us, after 7 rounds total, to have 8 acetyl-CoAs condensed together to make a C16 fatty acid. How do we complete this? Well, essentially you've got a C16 fatty acid attached to the acyl carrier protein. You do hydrolysis of this thioester and you get your free fatty acid. Pretty straightforward. Now those fatty acids can be assembled into triglycerides. As part of kind of our activity and maybe even a case study, we'll spend a little bit of time talking about the chemical triclosan, which is used as an antimicrobial, and the reason is it actually interferes with one of these fatty acid synthesis steps in a separate enzyme that bacteria have. So we'll spend a little bit of time talking about that, probably in a case study. Just like when we were dealing with beta oxidation, I didn't spend a lot of time talking about unsaturated fatty acids or odd chain fatty acids. I'm not going to spend a lot of time talking about how do we make all the different flavors of fatty acids. If you want to make it longer than 16 carbons, you use an enzyme called an elongase. You want to add double bonds to it, you use an enzyme called a desaturase. Just a couple of things to think about with those desaturases is we never put them, we're not able to put them at the delta 12 or delta 15 positions. Those are kind of those omega fatty acids, and double bonds are never put in conjugation. So not a lot of details we need to know from that, just some kind of big picture things to remember. We'll spend time in class talking a little bit about the details, but again, we've got our triglycerides here, or our phospholipids that we want to make. We've only made fatty acids at this point. So how do we put those onto that glycerol backbone? So some details for that, we can kind of go through in a little bit more detail with our activity. Again, we'll kind of look at a summary of this to remind ourselves about the big picture for this, and then that'll be it for our fatty acid synthesis.